Hi, my name is Sam, and this is our poster presentation on evaluating serum long non-coding RNAs as potential biomarkers for lung cancer. In this particular study, we went through and we developed some tools for using real-time PCR-based technology for identification of cancer-related long non-coding RNAs, as well as going through and developing out a new database to design our qPCR algorithm development. In this particular study, we're going through and we're comparing 3 samples from non-small cell lung cancer patients to 24 normal individuals. However, to go through and to look at a comprehensive list of long non-coding RNAs, first we needed to go through and start with a database. We decided to go through and look in the NCBI ref sequence as well as the Ensemble ENCODE databases for long non-coding RNA sequences to drive our qPCR assay design. In the end, we decided to go through and to actually merge both of these databases together into a comprehensive long non-coding RNA database in order to go through and to design long non-coding RNA qPCR assays. We went ahead and we took our qPCR assays and a step further, and we've laboratory verified every single assay that we're producing at this time. We go through and we qualify them for amplification efficiency, sensitivity, and specificity. After we have gone through and designed our qPCR assays, our bioinformatics scientists at QIAGEN went through and grouped them based on particular functions and on particular diseases. We've gone through and we've designed different panels of long non-coding RNAs. And in today's study, we're going to be using our cancer-related long non-coding RNA panel. We call this our Human Cancer Pathway Finder array. In this particular array, we have 84 long non-coding RNAs that have been associated with different types of cancers, everything from bladder, breast, cervical, and other types of cancers that you can think of, as well as incorporating 5 different normalization or housekeeping genes. 3 of these are traditional mRNAs, while 2 of these are small non-coding RNAs, and we have our patented control panel on the bottom of each of these arrays. These allow us to go through and to look for genomic DNA contamination, allows us to evaluate the reverse transcription efficiency, and also allows us to go through and look whether there's any inhibitors to the PCR process. Now that we've gone through and we've designed out qPCR assays for long non-coding RNAs, we've put them into our RT² long non-coding RNA PCR array system. We're ready to go through and detect serum long non-coding RNAs that are in our non-small cell lung cancer samples as well as our control samples. To do this, we went through and we started with 200 microliters of serum sample. We used the QIAGEN Mirror and Easy kit, which allows us to go through and quickly isolate all the RNA that we have within our samples. After we've gone through and we isolated all the RNA, we went through and we did cDNA synthesis using both random hexamers and oligo-DT in order to completely reverse transcribe long non-coding RNAs that may or may not have a poly A tail. We took this cDNA and we did a pre-amplification step. We did 12 cycles of pre-amplification using only the assay-specific primers that we're going to use and detect with our PCR array. After we've gone through and done the pre-amplification, we took the cDNA, mixed it with the master mix and loaded it onto our ready-to-use plate, put it in our favorite real-time PCR cycler and ran 40 cycles, and then we uploaded the data into our data analysis website. Here we have the results from going through and comparing the serum samples from our 24 normal and our 3 non-small cell lung cancer patients. This is a volcano plot that is summarizing the data. To the right here of this line are long non-coding RNAs that are upregulated. To the left of this line are long non-coding RNAs that are downregulated. This line here is a p-value line of 0.01. As you can see here, we identified several long non-coding RNAs that are differentially upregulated in our non-small cell lung cancer patients as compared to our normal patients. We've gone through and unblinded some of these, PVT1 and RMRP. These are long non-coding RNAs that are thought to be cancer associated, and here we're seeing the same sort of thing in our lung cancer patients. So in conclusion, we've gone through and we've developed out specific long non-coding RNA qPCR assays. In this particular study, we went through and we detected long non-coding RNAs in both the serum from 3 non-small cell lung cancer patients and compared this back to 24 normal individuals. In this particular study, we were able to detect 44 long non-coding RNAs, and we found several that were differentially upregulated with significant p-values in our non-small cell lung cancer patients. By going through and using simple real-time PCR technology, Coupled with easy RNA isolation techniques, we can quickly go through and start to screen more samples to see whether or not these potential biomarkers are consistent across more types of cancers and within lung cancer patients.
Yes, have you looked at exosomes?
When we went through and we started our project here, we started with serum samples. Our idea is to go through and look at all the long non-coding RNAs that are within serum, identify those that are differentially expressed, and then we can go back and use the Excel RNeasy kit and enrich for the exosomes and look for the long non-coding RNAs that are exosomal.
I understand that this data is from humans. Can you handle other species for link RNA analysis?
When we went through and we made our comprehensive database, we did this for both human and mouse. So we can go through and we can do the same sort of study with mouse samples.
Can we create our own link RNA panel?
We can go through and we can create your own panels and we can customize the format. Our custom panels can come in any 96, 384, even for the Fluidigm biomark. You can go through and you can use our comprehensive database to go through and select long non-coding RNAs, also include important mRNAs, and all the patented controls that we have in our qPCR system.